Hello, NEET 2026 aspirants. Are you stuck at 200, 260, or 280 marks in NEET? So, let's increase your score by minimum 100. Today is day 2 of our last 10-day series for NEET 2026. Today, we are going to cover the diagram based questions. First of all, we are going to start with the 11th class, and I am going to teach you the zoology part. So let's start with the living world. As you all can see, figure 1.1 displays the taxonomic categories. So first of all, we have kingdom, then comes phylum or division, then class, order, family, genus, and species. You can learn it by taking the first initial letters of each of the categories and by learning it as KPC of GS. So, as you move upwards, you need to remember one thing, that kingdom is the largest category. So, if it is the largest category, then it will have the maximum number of organisms. So, as you move from down to up, the number of organisms are going to increase. But one thing that will decrease is the number of common characteristics. Is that clear? Next, moving on from animal kingdom, look at this figure 4.1a. There are two types of symmetries, as you all know. First is radial symmetry, which is depicted in 4.1a, and 4.1b depicts bilateral symmetry. Bilateral symmetry is only one symmetry is possible, that is from the midline. Next, 4.2 displays the germinal layers. So, one is diploblastic and the next one is triploblastic. Diploblastic means there are two layers and triploblastic means there are three layers. A shows mesoglia, ectoderm and endoderm, and the B shows ectoderm, endoderm and mesoderm. Is that clear? Next, 4.3 shows A coelomate, B pseudocoelomate and C acoelomate. As you all know, coelom is the body cavity. So if you look closely, in A there is a true body, that is a coelom is present. In second, pseudocoelom is present, and in C there is no coelom present. Next, as you can see in the diagram 4.5, it is examples of porifera. First is cycon, second is euspongia, and third is spontula. You need to learn the names as well as you need to identify these correctly. Next, moving on, 4.6 depicts the examples of Coelenterata indicating the outline of their body form. First is Aurelia, that is Medusa. B is Adamasia, that is a polyp. 4.8, this is a commonly asked questions which is repeated several times. This is an example of Ctenophora, okay, that is Pleurobrachia. Next, look at figure 4.10. We have the example of Escalmenthes, that is the roundworm, male and female. Next, 4.11 depicts examples of Annelida, that is Nereis, and the second is Hirudinaria. Don't get yourself confused. Next is 4.9, which is the examples of Platyhelminthes. A is tapeworm and B is liver fluke. Next, 4.12 depicts examples of Arthropoda. A is locust, B butterfly, C scorpion, and D is prawn. 4.13 depicts the examples of mollusca. First is Pila and B is octopus. Next, if you look at figure 4.14, we have examples of Echinodermata, that is A is Asterias and B is Ophiura. If you look at figure 4.15, that is Belenoglossus. Now, the three main labels that you have to learn are proboscis, collar and trunk. 4.16 depicts chordata characteristics. Now, as you all know, chordata is the one which has a notochord. So the first labeling that is very important is notochord, then is the nerve cord, then are the gill slits, and then is the post-anal part. Now 4.17 depicts Ascidia and 4.18 shows a jawless vertebrate, that is Petromyzon. 4.19 depicts the examples of cartilaginous fishes. First is Scoliodon and B is Pristis. Now 4.20 depicts the examples of bony fishes. First is your hippocampus, B is Cutla. 4.21 depicts examples of amphibia, that is A is Salamandra and B is Rana. 4.22 depicts reptiles. First is chameleon, B is crocodilus, C is Chilon and D is Naga. Moving on, 4.23 depicts some birds. First is Neophron, B is Struthio, C is Pistacula and D is Pavo. Next, 4.24 depicts some mammals. First is Ornithorhynchus, B is Macropus, C is Pteropus, and D is Beliumtera. Now, moving on to the next, we have structural organization in animals. Figure 7.1 displays simple epithelium. Look closely. A depicts squamous cells, that are flattened cells. B depicts the cuboidal cells, which are cube-like cells. C depicts the columnar cells. And D depicts columnar cells bearing cilia. Next, 7.3 depicts the compound epithelium, which has multi-layered cells. Next, figure 7.2 depicts the glandular epithelium. A is unicellular, B is multicellular. Figure 7.4 depicts loose connective tissue. A is areolar tissue and B is the adipose tissue. Now, look in the areolar tissue, we have mast cell, we have macrophage, we have fibroblast and collagen fibers. You need to remember the identifications. In B, there are fat storage areas, nucleus and plasma membrane. Next, in figure 7.7 we have muscle tissue. A is the skeletal or striated muscle tissue where you can see the striations. B is the smooth muscle tissue. There are no striations. And C is the cardiac muscle tissue. It contains striations and there's also a junction between the adjacent cells. Moving on, we have figure 7.8 which depicts a neural tissue, that is a neuron with neuroglia. So you can clearly see axon, you can see the cell body with nucleus, and you can also see dendrite and neuroglia. Next, if you look at figure 7.5, you can see we have examples of dense connective tissue. A represents the dense regular and B represents the dense irregular. Next, figure 7.6 depicts the specialized connective tissues, that are A is cartilage, B is bone and C is blood. So in cartilage you can see we have collagen fibers and cartilage cells that are known as chondrocytes. In B, that is bone, you can see there are compact bone tissues and bone cells that are known as osteocytes. And in C, that is blood, you can see RBC, WBC and platelets. Moving on, figure 7.14 depicts the external features of a cockroach. So starting off, first of all we have the filiform antennae, then we have the head, then we have the compound eye, then we have the pronotum, then we have tegmina, we have the hind wing, we have the abdomen, we have anal cerci. Now we also have mesothorax, prothoracic leg, metathorax, mesothoracic leg and metathoracic leg. Next, now we have the breakdown of the head region of the cockroach. So A depicts the parts of head region and B depicts the mouth parts. So for the head region we have ocellus, compound eye, maxilla, mandible, labrum and labium. For the mouth parts we have labrum, hypopharynx, mandible, maxilla, labium, maxilla and mandible. Now mandible is divided into two parts, that is grinding region and the incising region. Moving on, look at figure 7.16 which is the alimentary canal of cockroach. Starting off with the pharynx, then salivary gland, then salivary reservoir, oesophagus, crop, gizzard, hepatic ceca, mesentron or midgut, malphizian tubules, ileum, colon and rectum. Next, 7.17 depicts the open circulatory system of cockroach. Starting off, first of all we have anterior aorta, then aliary muscles and then we have the chambers of heart. Moving on, reproductive system of cockroach. A depicts male and B depicts female. So starting off with the male, we have testes, we have phallic gland, small tubules, long tubules, seminal vesicle, vas deferens, ejaculatory duct, right phelomere, ventral phelomere, anal circus, caudal style, pseudopenis, titillator and left phelomere. You need to practice these by looking at them again and again and drawing. Next, we have breathing and exchange of gases, starting off with figure 14.1 which shows a diagrammatic view of human respiratory system, which is a sectional view of the left lung is also shown. Starting off we have epiglottis, then larynx, then trachea, then we have bronchus, cut end of the rib, lung, diaphragm and heart, as you know, is in the center. We have pleural membranes, we have alveoli, we have pleural fluid and bronchiole. Moving on to figure 14.3 where you can see a diagrammatic representation of exchange of gases at the alveolus and the body tissues with blood and transport of oxygen and carbon dioxide, where you can clearly see that inspired air comes in and the expired air goes out, the alveolar air. PO2 is 104 mm of Hg and PCO2 is 40 mm of Hg. Note the difference in these. Then you can see that pulmonary vein, pulmonary artery take the respective gases and bring them to alveoli. I want you to look at figure 14.3 that depicts the diagrammatic representation of exchange of gases at the alveolus and the body tissues with blood and transport of oxygen and carbon dioxide. Now the important question from this is the PO2 and PCO2 and what are their values in the alveoli and in the tissues. So many questions can be asked from this. Moving on, we have figure 14.2 that depicts the mechanism of breathing, showing A, that is inspiration, and B, showing the expiration. In A, you can clearly see that the diaphragm is contracted and the volume of thorax gets increased and the ribs and sternum are raised. Whereas in expiration, you can see that the diaphragm gets relaxed and it is arched upwards and the volume of thorax is decreased and the ribs and sternum return to their original position. Next, figure 14.4 depicts the diagram of section of an alveolus with a pulmonary capillary. Figure 14.5 depicts the oxygen dissociation curve wherein we have partial pressure of oxygen at the x-axis and we have percentage saturation of hemoglobin with oxygen at the y-axis. Next, we have body fluid and circulation. Figure 15.1 depicts the diagrammatic representation of formed elements in blood. Okay, so first one is RBC, second is platelets, third are your eosinophils, then next comes the basophil, neutrophil, monocyte, T lymphocyte and B lymphocyte. Now figure 15.4 depicts the schematic plan of blood circulation in human, that how blood gets circulated and how your oxygen and deoxygenated blood are removed from your body parts. Next comes figure 15.2 where we have a section of a human heart. Now you need to learn these labelings very nicely as to which part is aorta, which are the pulmonary artery veins, and you should also be knowing the labeling of bundle of His, intraventricular septum, apex and chordae tendineae and sinoatrial nodes. These are some important ones. Next, figure 15.3 depicts your standard ECG, that is PQRST. Clear? Now, next, we have excretory products and its elimination, starting off with figure 16.1 where we have human urinary system. So, first of all, we have adrenal gland, then we have the renal artery, renal vein, kidney, dorsal aorta, ureter, urinary bladder, urethra, cortex, medulla, pelvis and inferior vena cava. You need to learn the parts of kidney nicely. What are those? Pelvis, medulla and cortex, and which arteries and veins are coming out of them. Then we have figure 16.2 where you can see a longitudinal section of the kidney. So we have renal artery and renal vein which are entering into the renal pelvis, and we can clearly see the renal columns, the cortex, the renal capsule and the medullary pyramid. Okay. Next, 16.3 depicts a diagrammatic representation of a nephron showing blood vessels, duct and a tubule. You need to learn these labelings very nicely, and also the functions of collecting duct. It's a question that has been asked multiple times. Proximal convoluted tubule, distal convoluted tubule, efferent arteriole, what is glomerulus, Bowman's capsule. Then Henle's loop. It consists of descending limb of loop of Henle, ascending limb of loop of Henle, then below it comes the vasa recta, right? Next, moving on, we have figure 16.5 which depicts the reabsorption and secretion of major substances at different parts of the nephron. You need to understand what gets absorbed at the cortex and what gets absorbed at the medulla, or what is being secreted out. All of these things are very important. Next is 16.4 which represents the Malpighian tubule, that is the renal corpuscles. We have efferent arteriole, Bowman's capsule and PCT. Next is the diagrammatic representation of a nephron and vasa recta which shows the counter current mechanisms. Again, you need to learn the values and what substances are being absorbed or are coming out in outer medulla and in the inner medulla and in the cortex. Moving on, in locomotion and movement, figure 17.1 depicts the cross-sectional view of a muscle which shows the muscle bundles and muscle fibers. So first of all, we have a fascicle, that is a muscle bundle, which includes your muscle fiber, that is the muscle cell, sarcolemma and the blood capillaries. Moving on, figure 17.2 depicts your A part depicts the anatomy of a muscle fiber which shows a sarcomere, that is a complete unit, and B is showing sarcomere individually. Okay, so you need to understand where does Z line come, where is the H zone, where is the A band, where is the I band. Figure 17.3 depicts your A part depicts an actin, that is the thin filament, and B depicts your myosin monomer, that is meromycin. So A part depicts troponin, tropomyosin and F actin. B depicts your head, your cross arm, and we have two sites, which are those actin binding sites and the ATP binding sites. Now figure 17.4 shows the stages in cross bridge formation, rotation of head and breaking of the cross bridge. Okay, so as you can see, in first one actin filament where we have ADP and phosphorus coming in, which then leads to the myosin head forming the cross bridge. Then we have sliding or rotation. Then we have the breaking of cross bridge. Okay. Next is figure 17.5, which is the sliding filament theory of muscle contraction, that is movement of the thin filaments and the relative size of the I band and H zones. That what happens when you try and contract a muscle, and how does a relaxed muscle look, what does a contracted muscle look, and what does a maximally contracted muscle look. Okay, moving on. Figure 17.6 depicts your human skull. Okay, so you need to know which bones compose your cranium and which bones are present. For example, starting off we have frontal bone, then we have sphenoid, ethmoid, lacrimal bone, nasal bone, zygomatic bone, we have your maxilla, mandible, hyoid bone, parietal bone, temporal bone, occipital bone and occipital condyle. Moving on, we have figure 17.7, which depicts your vertebral column, and this is a right lateral view. Starting off with the cervical vertebra, then we have the thoracic vertebra, then we have the lumbar vertebra, and then we have the sacrum, and then we have the coccyx. And you can also see the intervertebral disc. Next, figure 17.8 depicts your ribs and rib cage. So, in the center we have your sternum, then we have the ribs, then at the posterior side we have your vertebral column, and as you all know, 1 to 7 are the true ribs, then 8 to 10 are your false ribs, and 11th to 12th are your floating ribs. These are also important. Figure 17.9 depicts the right pectoral girdle and the upper arm, and this is a frontal view, starting off with clavicle, then we have the scapula, then humerus, then radius, ulna, carpals, metacarpals and phalanges. Don't get confused in carpals and tarsals. You should know that carpals, metacarpals and phalanges are present in your hand, and your tarsals, metatarsals and phalanges are present in your leg. Figure 17.10 depicts your right pelvic girdle and lower pelvic limbs. This is also a frontal view, starting out with the ilium, coxal bone, sacrum, pubis, ischium, femur, patella, tibia, fibula, tarsals, metatarsals and phalanges. Alright, I hope this is clear. Moving on, we have neural control and coordination. So figure 18.1 depicts your structure of a neuron. Starting off with dendrites, then we have the nasal granules, which you can see as tiny bodies present in it. Then we have cell body, nucleus. Then we have the Schwann cells, axon, myelin sheath. Then we have the nodes of Ranvier. Moving on to figure 18.2, where we have the diagrammatic representation of impulse conduction. You need to see what influx is happening. Next, we have figure 18.3, which shows the axon terminal and synapse. Starting off axon, then we have the axon terminal, then we have the synaptic vesicles, then we have the neurotransmitters, and we have the presynaptic membrane, we have the synaptic cleft and postsynaptic membrane, and these three together form your synapse. Okay, and then we have the receptors. Moving on, figure 18.4 shows your diagram which shows the sagittal section of the human brain. First of all, in forebrain we have cerebrum, thalamus and hypothalamus. And you can clearly see the cerebral hemisphere, you can see the corpus callosum, you can also see the cerebral aqueduct. Now moving on, after forebrain we have the midbrain, then we have the hindbrain, which consists of pons, cerebellum and medulla, and below it comes spinal cord. Now the next chapter is chemical coordination and integration. Figure 19.1 depicts your location of endocrine glands. Okay, and what are those? Starting off first, we have hypothalamus, then we have the pineal, and then we have pituitary. Then we have thyroid and parathyroid glands. You also need to know the numbers of it. They are going to try and confuse you as if how many parathyroid glands are present, and you should also know the number of pairs. Then comes thymus, then comes your pancreas, adrenal glands. In males, we have testes, and in females, we have ovaries. Okay. Now, figure 19.2 is a diagrammatic representation of pituitary and its relationship with hypothalamus. So, first we have hypothalamus, then we have the hypothalamic neurons, which have an innervation in your posterior pituitary, and we also have the portal circulation. We have anterior pituitary and we have the posterior pituitary, which combine up with the hypothalamus. Is that clear? Then we have figure 19.3, that is the diagrammatic view of the position of thyroid and parathyroid. So A is the ventral side, okay, where you can see we have vocal cords and we have thyroid and we have trachea. In B, that is the dorsal side, you can see 4 small parathyroid glands, as I was talking about the number of pairs, okay. Figure 19.4 depicts the A depicts adrenal gland. It is present above kidney, that is why it is also known as suprarenal gland. B is the section showing two parts of adrenal gland. Okay, so first, as you can see, we have kidney, over it we have the adrenal gland, and adrenal gland is divided into two parts. And what are those? Adrenal cortex, that is the outer part, adrenal medulla, that is the inner part. Next, as you can see, figure 19.5 is a diagrammatic representation of mechanism of hormone action. So A is the protein hormone and B is the steroid hormone. So example, FSH, how that hormone comes in the receptor, which is in the ovarian cell membrane. Then we have response 1, which leads to the generation of second messenger, that is the cyclic AMP or calcium. Then we have biochemical responses, and then we have the physiological responses, that is, for example, ovarian growth. Now, for steroid hormone, example estrogen, we have the uterine cell membrane and this hormone is going to cross it. Then it leads to formation of a hormone receptor complex. We have the nucleus that leads to, which further leads to genome, mRNA, proteins and physiological responses, that is the...
Hello NEET aspirant, I'm gonna be helping you out with your botany 1 quick 11th class all diagram revision. Let's just start with it. The first chapter is the biological classification, where it has divided the whole chapter into different kingdoms. So the first kingdom is the kingdom Monera, where first thing is what their shapes has been given to you, that is cocci shape, bacillus shape, spirilla shape and the vigorous shapes. So these are the 4 shapes. Then for the eubacteria, you have a special example nostoc given to you. Very important is the labeling, that it has a mucilaginous sheath around it. So means it is present in the aquatic environment. And second thing is that there is a presence of heterocyst cells, which is the special cells which help in the nitrogen fixation. And this is also its biofertilizer used as that feature. Then it has shown the binary fission dividing of the bacteria, how they reproduced. Moving on to the next kingdom, that is a protista. Protista, we have total 5 different division into it, but 4 diagrams are being given to it. Number 1 is the dinoflagellate. Cryophytes, no example given to you. Sorry, no diagram example given to you. Then you have the euglena. Then you have the slime moth. Slime moths are what? When they comes together and form a big structure. This is being shown to you, that Plasmodium. Then we have the protozoan ones, where the paramecium is being shown to you. After that, moving on to the next kingdom present is the kingdom fungi. In fungi, 3 diagram. Number 1 is the mucor. Most probably this diagram won't come in your paper because it's a very vague diagram. Then comes this comes to you as a asparagus, and what you are seeing is the conidia over there. And then you have this agaricus. Next, The final is the place which was like R.H. Whitaker did not give any place to these organisms. They are what? Virus and viroids. So virus examples are given to you. This is what? This is the tobacco mosaic virus. It has two things. One is the RNA, which acts as its genetic material, and the second is the caspid, that is what? Its protein code. Very important that this virus told us that RNA can also act as a genetic material. Moving on to the next one. This is the bacteriophage, means those virus which infects the bacteria. It has proper labeling of head, collar, sheet, tail pins, all those things which you have to remember. Moving on to the next chapter, that is kingdom fungi. Kingdom fungi is divided into 5 different divisions. Number 1 is the algae. All these algae are being shown to you. Algae is further divided into 3 different classes. So this is what? Chlorophyceae. This is the rhodophyceae, and sorry, this is the phycophyceae, brown algae, and this is the rhodophyceae. In the chlorophyceae you have volvox, which act as a colonial form. This is a long filamentous form of the eulothrix. Then in case of brown algae, you have what? You have these 3 examples. Very important is that in this example, that is the laminaria, it has air bladder which provide its buoyancy. Coming back to the other examples, that is the first one, that is your brown algae. This is your, sorry, the first one is your first one is the laminaria, second one the bladder, that air bladder has the fecus one, and the last one is the dictyota. Then we have the red algae. 2 examples are given to you. Number 1 is porphyra, and the second one is the polysophonia. Moving on to the next, that is the braphites. In braphites you have the example, the first examples are your liverworts, because they look like a liver form. So here you have the example of marchantia. Very important thing is that it is showing what? It is showing the male and the female part. So you have to remember that the male part look like this, female look like this. The male part special terminology are antheridiopore is being given. The female is the archegoniophore. Then you have to remember this male and the female part. Moving on to the next thing, that there is what? There is mosses also present. So 2 such examples are given to you. The number 1 mosses is the funaria. Number 2 is the sphagnum. Sphagnum is specially used as a peat. Okay, so this thing has economical importance also. If you see over here, you can see in this proper, you have what? Sporophyte and the gametophyte part. So, and the first one, this which I have showed you about the marchantia, they are 2 different bodies, so means they are dioecious. Okay, this thing also you have to remember. Then the sporophyte has 3 part, capsule, seta, and the foot, which is not being shown. Okay, and whereas gametophyte has the leaves and the leaf-like or root-like structures present. Moving on to the next one, that is pteridophytes. The first one is the Slegionella. This is the equisetum. Very important, in equisetum you find the strobila. Strobila is like cone-like structure. Then this part is this one is the ferns, and these are the salvinia. Moving on to the next one, we have angiosperms. The first one is the cycas. A lot of example of cycas are being given to you. Its characteristics are given to you. Those you have to remember. This is the pinus, and this is the ginkgo that the diagram is given to you. Moving on to the next one, you have what? You have your angiosperms present. 2 divisions or 2 classes are present of the angiosperm, that is dicot and the monocot. You will say that from this diagram how we come to know that monocot has upright leaves, that are what? They are what? Isobilateral leaves. So the leaves are in the isobilateral position. That's how we can distinguish it. Moving on to the next, morphology in the flowering plants. So your whole angiospermic plant has these structure. It has a shoot system and a root system, and the roots are normally the primary roots which will be there, and then from here, if it's a dicot plants, there will be secondary roots present to it, or the lateral roots present to it. But if we see towards the shoot part, we see what? We see the internodes, nodes, and the leaves and the flowers which are present on it. Now, one by one, we will see the vegetative part and then the floral part of it. In the vegetative part, this is the stem part. So, roots part. The roots could be of 3 types. One is what? These are the tap roots, fibrous roots, and the adventitious roots, which are coming from the other part. The root has 3 sections. One is the region of meristematic, region of elongation, and region of the maturation. Region of maturation only has the root hairs present, and the region of meristematic tissue has a covering called root cap. Moving on, the roots can have modifications. So these are the example of the root modifications. What are those? Number one here is called the asparagus, where the nodules kind of roots are present. Then we have what these storage roots present in the form of turnip, carrot, and sweet potato, depending upon its shape and size. So it could be circular, it could be conical, it could be tuberous. And then we have what respiratory roots, which is seen in what nematophores. Sorry, as the nematophore seen in the your mangroves plant. Then we have this modification in the stem part. So either the stem can act as a storage part, like in case of ginger and all those examples. Then we have what the stem can act as a tendrils for the climbing. Then the stem can have a modified into spine, so with that will help into what in the protection. And it could be offsets, like it grows from one part and goes like over the water, over the soil or under the soil, and run through it and comes out as a new plant. Okay, so this could be there. Then comes to the leaf part. The leaf is what this structure, where it has this whole body called as lamina. It's this margins are there. This is the apex of the leaf. This is the petiole part through which it is attached to the main axis. It has stipules present over there, axillary bud present from which other things can come out of it. Then the leaves could be of two different types in terms of veneations. Veneations type is what? That either it could be network forms, reticulate, or it could be parallel form, which is parallel to the midribs. Then the leaf could be of two types. It could be of simple leaf or the compound leaf. Simple leaf is what which we have seen it. Compound leaf means this sideway incision goes up to the midrib and each make a different leaflet. This could be of two types. It could be pinnatately or palmatedly. Then comes to the next thing, that how the leaves are arranged on the stem, that is known as phyllotaxy. It could be of three types. One is alternate, one is the opposite, and the one is the whorled, from where from single node multiple leaves are present. Then the flowers can be arranged in the form of on the stem, and that is called as what? Inflorescence. There are two types of inflorescence, racemose or the cymose. In cymose, you can see that once the stem will grow, it will have the older flower, and lowermost will the new smaller plant will keep on coming up. Then comes to the next thing about the flower. In the flower, how that all four worlds of the flower is present, this different categorizations or the classification has been there. Now in this, remember this here over here you have on the position of the ovary. If the ovary is present at the similar level, we call it what? Sorry, not the similar level. If the ovary is present above and all the other part is coming from the down, we call it what? Hypogynous. If everything is like something is present, some part is present above, some part is present below, this condition is called as what? Perigynous. And if the ovary is down and all the other parts are coming up above from it, is called as epigynous. Comes to the flower has what? These three parts: calyx, corolla, androecium, and gynoecium. How the calyx and corollas are arranged, we call it estivation. And estivation could be of these four types: velvet, this is twisted, this is imbricate, and this is the vexillary. Then the next one is the placentation, how the ovule are attached or present inside the ovary. It could be marginal, it could be axile, it could be parietal, free central, and the basal. Okay, moving on to the next one, that is that your this thing will change, like your ovary will change into what fruit. There are true fruit examples given to you, and that are what? Mango and the coconut. Its covering, outermost layer is the epicarp, which is fleshy in case of, not the fleshy, but it's a thin covering in case of mango, and it's fibrous in case of what? Sorry, it's also a covering, normal covering, hard covering. Then mesocarp in case of mango is the pulpy one, fleshy one, whereas in the coconut it is the roots, fibrous one, hairy ones. Then the seeds, and before seeds there is what? White color part, or in the case of coconut it's a hard shell kind of a part that is present, which you broke up and get the white color cream out of it. Then the seeds could be of monocot or dicot. Here the monocot seed and dicot seed is given to you. In dicot seed you can see that two cotyledons are given, plumule and radicle is given to you. Seed coat is also being shown to you. Hilum and the micropyle is also shown to you. This you can easily link up with the sexual reproduction in flowering plant as well. When it comes to the maize has a special thing that they have shown to you, that is what? Endosperm present, and endosperm is covered by the alluron layer, that is what? Your proteinaceous layer, and this is your embryo part of it, and its labeling giving to you. After this, in this chapter, you have what? The floral diagrams. In floral diagrams, you have to remember every part is showing certain kind of a whorl arrangement. The outermost is showing what? Your calyx. This one is showing your corolla. This one is showing your androecium, and the innermost one is the gynoecium. You can see that whether you can cut it into any half, one half, two half, or multiple half, you can write it about its zygomorphic or the ectomorphic category. Male, female, both are present. You can call it bisexual flower, and you can simply count them and write them. On this basis, these three families are super important, out of which this is given in new NCRT also, whereas about the Solanaceae family, this is given in your NCRT, but these two family, that is Fabaceae and the your Liliaceae family, is not given. So this you have to remember from the old NCRT, has been marked over here. You can take a screenshot and study about it. Okay, then the next thing comes to the next chapter, that is anatomy in flowering plants. So now in this chapter, you have what? Certain parts which are not available in the new NCRT. So these are those, the parts. Number one is what? That these are what? Different meristems, apical meristem showing. This is the root meristem. This is the shoot meristem. So if you see that it has a root cap, it is the area, it is a zone which is what? Highly having the cells which are dividing. Okay, and then you can see that there is what? There is a central cylinder which has what? The xylem and phloem that will be present. These are the cortex area, and this is the epidermis present to it. Okay, so these are certain labelings given to you which you have to remember. When it comes to shoot part, very important, there is what? Axillary bud present, shoot apical meristem. From here what? A leaf comes, so shoot apical meristem zone, and the leaves are coming from it, and the xylem phloem which are present over it, which has to keep on going to the all parts accordingly. Then comes to the types of tissues other than meristematic tissues, that is what? Permanent tissues. So you have simple permanent tissues and the complex permanent tissue. In simple, this is parenchyma, this is collenchyma, this is sclerenchyma, two parts, sclerids. Sorry, this is the sclerids and this is the fibers. When it comes to the vascular part, you can see that this is the xylem and this is the phloem present to it. Xylem has four parts, two are being shown over here, that is tracheids and the vessels. Whereas in the phloem tissue, they have shown you almost everything, that is sieve tube elements, phloem parenchyma, companion cells, only the fibers are not being shown over here. Coming up to the next thing is the tissues present over there. So you have what? Epidermal tissue. These are the two different types of what? Two different types of your stomatas. Number one is this dicot stomata, which is kidney bean shaped, and this is dumbbell shaped stomata present in case of what? Your monocots. Here these labelings are given to you, very important. These are what? Guard cells. Okay, these around this guard cells, what are there? These are the subsidiary cells. Okay, subsidiary cells, sorry, these are the subsidiary cells, this one, which help in like keeping it the guard cells safer. Okay, these things are there, and the center part is what? The stomatal pore through which the transpiration takes place. Then you have what? Tissue systems, which are, sorry, the organization, anatomy of this. This is roots, this is stem, and this is the leaf. Starting with the roots one, if you see that what's the major differences, you can see that here is only four xylem part that is being shown. So it is what? It is tetrarch kind of conditions, whereas over here this is more than 6 present. So if more than 6, that is polyarch condition present, seen in case of what? This is monocot, this is dicot. Rest, if you are going from outside to in, what you will see? You will see the epidermis in both the cases, you will see the cortex, then you will see the endodermis, then there is the pericycle. Going inside, you have this area which has been what? Alternative parenchyma and the xylem phloem present. The xylem phloem present in the center is called as pith, whereas over here it is called as conjunctive tissue. And in between these area and certain areas over here is showing what? Xylem and phloem. Their arrangement is what? Their arrangement is a radial present over here. I think one diagram is missing from here. Okay, yeah, this I forgot. So yeah, this is the vascular bundle arrangement, how they will be present. So in the roots, it's the radial. These are what? This is the xylem and this is the phloem present at two different radii. Whereas in the stem, it is present two together. So they could be conjoint in these cases. And in these two cases, this is what? This is closed because there is no cambium present, seen in case of monocot stem. Whereas this part is what? It is open because it is open for the secondary growth, that is cambium is present, which is a dicot kind of a one that is shown to you. After this, two things which you see over here. Next is the stem part. In the stem, very important is there the monocot. So this is the dicot, and this is the second one is the monocot stem. How different they look like, what are the layers present to it. Very important is that if you see the pith over here is highly developed, very big, very large. Here pith you can't even appreciate. So over here, what scattered xylem and phloem bundles present throughout the ground tissue area. After ground tissue you can't see anything, whereas over here you can see all the layers one by one properly. Very important is that what these layers are made up of. Over here if you see that the hypodermis is made up of collenchyma, whereas hypodermis over here is made up of sclerenchyma. Sclerenchyma is present over here in this area as well. So don't think that sclerenchyma is only present over here, but with respect to hypodermis I was talking. Moving on to the leaf part, this one leaf which is showing two different types of cells. One is this placid cells and the second one is these spongy cells. This is seen in case of dicot leaf. Moreover, on the dicot leaf, you will see the stomata on the lower surface, which is what abaxial surface. Upper surface is the adaxial surface. Then it comes to the monocot. You have what? You have both side, upper and lower. There is stomata presence. Equal thing. So to protect themselves, they have bulliform cells. With this arrangement, the next chapter is the cell chapter. In the cell chapter, this is the plant cell, this is the animal cell. You see this. Normally question don't come directly from the diagram of this, but we won't be taking any charges. So see this carefully and what are the parts present to it. If you have seen all the organelles, if you have studied it, this is much easier to understand. Coming back to the major thing, this is the certain cells present to you. These cells which are being present over here is the eukaryotic cell. This is what? Red blood cell. This is white blood cell. This is the columnar cells which are narrow, straight, and keep on going like this. This is the nerve cells. This is what? This is your tracheids, which is what? Your xylem cells. Okay, and this is your mesophyll cells, which is one the spongy ones present over here. Then you see your prokaryotes. In prokaryotes you can simply see that there is what? This is like, this is a eukaryotic cell, this is a prokaryotic, and their size is given to you and the virus also given to you. So typical bacteria 1 to 2 micrometer, pplo, that is 0.1 micrometer, whereas in the text it was written 0.3. so there is a bit confusion over here. Then viruses is much smaller than that. Then comes to the cell membrane. So this is your what? Your cell membrane, or called as plasma membrane. In this you can see that this thing is what? Lipid membrane present, bilipid layers. Okay, then in this you have what? These are the proteins, integral proteins. There are certain external proteins which are present outside, which is called as peripheral protein. Certain over here proteins are also present. Certain cholesterol molecules can also be seen over here. And very important is that the presence of sugar over here, which is making what? Your different types of receptors. Moving on to the all the possible organelles that left out in the chapter. Number one, this diagram is showing you what? Your endoplasmic reticulum. This is the smooth endoplasmic reticulum. This is the rough endoplasmic reticulum. What is specially present on the rough endoplasmic reticulum is the ribosomes on it. Whereas this area has what? Nucleus and the nuclear pores seen over here. This both organelles, smooth or rough, these are just the types of it. They are directly attached or in continuation with the nuclear membrane, which we can see. Then we have what? Golgi body, which started from just links to it because it is working in association with it. Over here you can see what? Cisternae. These things are what? These are the cisternae, and these are what? These are the vacuoles that are coming out of it. Vesicles coming out from it. Moving on to the next one is the mitochondria. In mitochondria you have double layers, outer membrane, inner membrane. Inner membrane is folded into cristae. So this is cristae, this is cisternae. Very important, the differences you should know. Then in between this area is the matrix, and in between the outer and the inner membrane, that area is called as intermembrane space. Then the chloroplast is also there. Chloroplast has what? Same has outer membrane, inner membrane. It has these structures which are made up of multiple thylakoids joined together called as grana. They are joined to each other by the stroma lamella, and the other area is called the stroma. Moving on to the next part, that is our cilia and the flagella. Cilia and flagella has these kind of structures. If you count, these are 9 and they are present in the pair. So 9 plus 2 arrangement is there, where 9 are this and 2 are these. But these 9s are in the doublet form and these 2 are single if you can see it. So these things and their markings are given, which you have to remember. This can come to in your paper. Then in the ribosomes, ribosome completely look like this. This is the larger and the smaller unit which join together with the help of magnesium ion. Moving on to the nucleus. In the nucleus, this is not very clear one. You will get a clear example where you have the nuclear membrane. You can see the genetic material. You can see the nucleoplasm. You can see this area which is called as nucleolus present over here, which is the factory of the ribosome, which is present over here. Then inside this, during the division time, you can see the chromosome. In the chromosomes, these are what? These are the arms of the chromatids. This is one chromatid, two chromatids. In the center, there is a constriction called as centromere, which has a proteinaceous coat called as kinetochore. Through this, the spindle fiber joins during the time of cell division. If you see the chromosomes according to its morphology, depending upon where their centromere is present, could be of 4 types. This is metacentric, submetacentric, acrocentric, and the telocentric ones. Okay, moving on to the next chapter, that is cell division and cell cycle. You can see that cell has what? 2 different phases. One is this interphase, other is the M phase. Interphase is G1, S and G2, and then start the M phase. M phase has 2 parts. One is the karyokinesis, second is the cytokinesis. In karyokinesis, we have what? Prophase, metaphase, anaphase, telophase, and the cytokinesis, and then the 2 cell forms. Sometimes within the G1 phase, the cell don't want to go ahead into another division, goes into inactive state called G0 stage, where it is metabolically active but not dividing or not making anything for it. When it comes to the M phase, you have the early prophase diagram, late prophase diagram. Then from late prophase diagram, it is changing into metaphase. The metaphase is the phase where every chromosome is arranged into the like what we say that into the central place called as metaphasic plate, and these 2 are what? Your centrosomes, or you can call it centrosomes, which are forming the estrol race and microtubule apparatus. Okay, then this metaphase will change into anaphase, where the splitting of chromosome will take place and that chromosome number will increase over here. Then comes the telophase, where what happened? The nucleus membrane again forms similarly to it, so you can say that, and it start dividing, and the cytokinesis will divide completely to it. Okay, then comes the meiosis. Meiosis is what? It's a 2-step process. Meiosis 1, meiosis 2. All these diagrams have been shown to you. By the end of meiosis 1, dyad of cell forms, while the end of the meiosis 2, you can see that tetrads of cell are being formed. You can see what that here there are 2 metaphasic plate. Okay, this is the very important point. So it shows the metaphase 1. Moving on, you can even see that there is what? Crossing over has taken place. So these things you have to remember, and by the end of this is what? Homologous chromosomes separate, not the centromere split or something like that. But over here again the centromere split like the mitosis. Moving on to the plant physiology, where we have what? We have photosynthesis, where this is showing your what? Showing you the experiment of the Presley that oxygen is important, that good gas is the oxygen. Then again this is a diagram of your 2D diagram of your chloroplast. Same things are given over here, which we have studied over there. Then comes the light reaction center. In the light reaction center, this is what? Your pigments, whole pigment present to it, and it takes up all this whatever the energy will come, that will excite what? Your electron, and it will go to the primary acceptor. Basic cycle start has been shown to you. Then comes this very important example about the absorption spectrum and the action spectrum. So what happens is that this is a different pigments and how their peak is very important to remember that. From that, for the chlorophyll A, they have taken up this peak. Okay, and in that peak you have seen what? They have superimposed it. So they see that from where the wavelength has been up, like most amount of light has been absorbed, the same area is what? Maximum amount of action has been taken, means oxygen is being liberated. So this is showing what? Action potential, this is showing what? Absorption potential, and this is showing what? The proper superimposition of that two things. Very important, remember it. Moving on to the next one, that is the light reaction and the dark reactions and the factor affecting it. So this is what light reaction, and this is what two types of dark reaction that is taking place. In the light reaction, if you know that in the inner mitochondrial membrane this thing is taking place, that is called as Z scheme. So your one photosystem 2, what will do, when the light will come, it will excite the electron, it will be taken up by the primary acceptor, and then according to its potential it will keep on coming back to its reduced unexcited state. While coming up, it is taking place with the help of lot of electron transport that is being taken place, has been shown over here in the chemoosmosis hypothesis also. Then from this it comes to the photosystem 2, which also get excited by when the light of 700 nanometer is coming up, it excites, electron goes over here and it comes. While coming down here here, it makes ATP and NADPH. This thing, now the very important thing is that in the stroma lamella has only photosystem 1 present. So photosystem 1, that is 700 nanometer one, it get excited, electron goes to it, but the electrons comes back to it only because over here the cyclic respiration take place, so cyclic photosynthesis take place, and in that time when the cyclic photophosphorylation is taking place, the ATP is only forming, means no NADPH will form in the stroma lamella. So this has been two things that we've shown. This is what cyclic, this is the non-cyclic photophosphorylation. Then this is the what? This is your chemoosmotic hypothesis, which is showing that inside there is hydrogen atom which is increasing, and in the stroma hydrogen atoms are reducing, protons are reducing. So what will happen? It creates a gradient. There is already a membrane, there is already a photosynthesis pump. So what will happen? The hydrogen will go from inside towards the outside, and this will create what? ATP over in the stroma, which will be used in these two reactions. Now there are two different types of plants, C3 plants, C4 plants. In the C3 plants, the three steps process is what? There is carboxylation, where is what? Fixing of carbon. So carbon is fixed with this 5 carbon compound, forms a 6 carbon unstable compound, which easily get degraded into 2 3 carbon compound, call it 3PGA. Now this 3-PGA undergo the reduction, means the aldehyde will change into what? This PGA is what? Acid. Acid will change into what? Aldehyde. So this is what? Reduction. Here the ATP and NADPH will used up, and over here again the regeneration will take place. Here also one ATP will be used up. This is the Kelvin cycle or C3 cycles or take place in the C3 plants. Whereas in C4 plants, what will happen? They have two different cells which are working into it, where this whole thing is taking place in C3 in the mesophyll cells. Now what will happen? That first the carbon dioxide will be taken up by with the help of what? For PEP, a 3 carbon compound that will make a 4 carbon compound, oxaloacetic acid, will change the malic acid and transport it inside this bundle sheath cell. Inside bundle sheath cell there is rubisco present. So what will happen? This 4 carbon compound will break down into 3 carbon and the 1 carbon. 1 carbon will go into the Kelvin cycle similarly to this, whereas the 3 carbon compound goes back over here, regenerate itself into what? The one which will be capturing it. So this thing has been shown to you. This diagram about which cell which is doing, very important. Next thing is that there is a factors affected, where the light intensity diagram is given to you, that as we keep on increasing the lighting, first the rate of reaction will increase significantly, and after that it becomes stable. So someone will ask that why this becomes stable over here? Because of the other factor which will become limiting over here. With this, the photosynthesis chapter is quickly being done. Let's move forward toward the respiration in plants. Respiration in plants are more about the flowchart based chapter. So here the first thing is what? Your glucose cycle, which is a 10-step process forming what? ATP, which takes place in all the organisms. Then comes the two different things, whether it's the aerobic respiration or the anaerobic respiration. If it's the aerobic respiration, then the pyruvate which is formed by the glucose during these steps will go into what? Change into acetyl coenzyme and undergo the cyclic the tricarboxylic acid cycle, where these different compounds, these different energies will be forming up. Take time, do through this, go through this, super important. These reactions can be coming to you as a step based, sequence based question also, so very important for you to go through it. These are not diagrams which I felt that these should be told to you in this part only. But if you are like, if something is it undergoes fermentation, but fermentation can only go by bacteria. No, it can undergo by certain eukaryotes also, like fungi undergo through it, and even certain other organisms can do that. Okay. Then in the fermentation, two different ways are there. Either pyruvic acid, which make the lactic acid, lactodehydrogenase it will use, or it can make the ethanol and the carbon dioxide. Depends upon it reaches very less amount of energy when the fermentation take place. Then comes to the in the aerobic, what will happen is that since there is NADH is also being formed as FADH2 is being formed as, which is what high currencies has to be converted into what ATP form. So it will go to the electron transport system, which is present on the inner mitochondrial membrane of the mitochondria. Now here these different complex are present. Complex 1 is this, complex 2 is this, complex 3 is this, complex 4 and complex 5 is this. Their names are given to you. Next thing that you have to remember, in the complex 1 what will happen, your FNM will take up once the like complex 1 is like when NADH is being converted into ATP, then first complex, third complex, fourth complex and fifth complex will work. But when the FADH2 will work, then all these second, third, fourth, fifth will work for the FADH2. So now what will happen, this thing will be broken down. The hydrogen as well as the electrons, they will be taken up by FNM. Hydrogen will be thrown into intermitochondrial space, where the main constant is what, to increase the content of the hydrogen, so that over here if the hydrogen is more, we can easily take it on this side and make the ATP, which has to be done. So this is what they are doing, it breaking up themselves and creating the concentration gradient, same as what chemoosmotic hypothesis. Now this FNM will go and form give the electron to FES, which will give to the carrier, that is, which is intermembrane carrier, that is ubiquinone. Ubiquinone simultaneously will take up the hydrogen, and that hydrogen it will use to transfer the electrons to the next complex. So if it is a third complex, of course in NADH2, NADH, that there will be what, third complex will work. So third, it gives the electron to the third complex and the hydrogen will again thrown out. So this way what, hydrogen one side is being thrown or dumped up. Now this cytochrome B will give it to FES and then cytochrome C1, and that's why the name is also there, cytochrome BC1, complex number 3. Now this whole thing goes to the cytochrome C. Cytochrome 3 give it to what, it's to give it to complex number 4, where it goes to copper A, then cytochrome A3, then the copper B, and then finally this hydrogen will come inside, as well as the electrons will also be dumped over here, and the oxygen will take it up and forming what, water. Okay, means hydrogen over here, how they will come it directly over here. Now the hydrogen is initially going out, so it comes inside over here. This hydrogen concentration increases, which goes and forms what, water. Simultaneously it is making ATP with this help of this ATP synthetase. This whole structure is given to you, how they are present. This is very important for you in the chemoosmotic hypothesis. The head is towards the stroma side, over here it is towards the matrix side, whereas this F0 is completely embedded in the membrane in the both the cases. So remember this thing. Then comes what, in except carbohydrate goes with this cycle. What about if fats and the proteins are being taken up as a respiratory substrate? Then they add up over these points, which you have to remember. With this, the respiration in plants are over. Next comes the plant growth and development. So over here the first diagram is showing that how the plants grows, where the very important thing is the presence of cotyledonous leaf initially that is present. Then the growth is what, growth take place by two, shoot growth, or like there are many ways of growth. If you remember the meristemic growth activities, how they take place, the shoot apical meristem is there, root apical meristem is there, lateral meristem growth is also there. So those parts which has been removed in that is actually being told over here. So nothing is being removed in reality. Then if you have to measure it, this is the one way of measuring it up, by pointing the markers, and after, as the sprout will grow, it will increase the length. So increasing the length, increasing the weight, increasing the water content, these are all ways of measuring the growth. Then this is the different ways of growth characteristics that you can find. If you see this leaf diagram, there is what, two things you can come to know, that real growth and the apparent growth. So if you see that real growth is actually the 5 centimeter square, but the apparent to it in this case, first one is 100% growth. Here only 10% growth will feel. So how this is what, the growth curves can be of two types, linear growth or the geometric growth. Linear growth is what, it is what, arithmetic growth, limited period of a time that is being taken place. But linear means equal intervals of the time, equal thing has been adding up. Whereas in the geometric growth, what is instant exponentially phase will come, but it can't sustain for long, because everything has a limitation that it will slow down, reduced or completely obliterated. Different diagrams has been given here. This diagram very important. It is showing that the zygote first has a geometric growth, increase the number of cells, and once it is increased, then it reduces it and changes into what arithmetic growth, where division is taking place but the functions are also being taken up. Then comes what? The plasticity. There you have seen what? Adult and the juvenile phase of the plant, and the terrestrial and the water environment differentiations. You can see that is a heterophily for the developmental and the environmental thing. Then over here this has been given what? Apical dormancy. If we remove this tip, we can let the plant grow further, so we can grow them further, other parts will grow. So the apical dormancy thing has been shown, and this diagram is showing what? That how the coleoptile moves towards the sunlight under the influence of the auxins. With this, your 11th class botany all diagram is quickly being revised to you. I hope you will remember these diagrams on the D-Day.